Thank you, everyone, for being here and for adjusting to the change in the venue. It's my pleasure to welcome Laura Rodriguez. She is an organic chemist from the Lunar Planetary Institute in Houston, Texas, and is also a visiting scientist here at UT through the center, working with various folks on some of the work that she's going to be talking about today. So her research is centered on understanding how planetary environments can drive the formation of genetic precursors on prebiotic worlds, and she's also interested in developing strategies to help elucidate the organic content in spectra acquired by planetary missions, astromaterials, and prebiotic analogs, where the abiotic chemistry can get very complex. She is an MSL participating scientist and a member of the MSR Campaign Science Group. She received her bachelor's in geochemistry from Rice University and a dual title PhD in geosciences and astrobiology from Penn State, and completed a postdoc at NASA JPL in the Origins and Habitability Laboratory. Thank you very much for being here.
Yeah, thank you for having me. Just doing a sound check. You can hear me still?
Loud and clear.
Okay, great. Thank you. Hi, everyone. So yes, my name is Laura Rodriguez, and today I'm going to be talking to you about strategies that I've been developing to hunt for organic carbon on planetary bodies. I've divided this talk into 4 sections. The first is going to be talking about trying to convince you all about the importance for prebiotic chemistry for planetary missions, some of the challenges that we encounter with prebiotic and abiotic samples, and thirdly, some machine learning approaches I've developed to try to facilitate the characterization of organics in planetary samples. And then finally, we're going to go a little bit of a left turn and talk about the proposed work, the work that I proposed for the UT Center for Planetary Systems Habitability Visiting Scientist Grant that I was awarded. So first, let's begin about prebiotic chemistry. So oftentimes when we talk about prebiotic chemistry, it can be thought of more as relevant for trying to understand the origins of life on early Earth. But the fact of the matter is that you can extend abiotic chemistry to learn something about the origin of life potential on other planetary bodies. And in fact, that can be critical in the search for life, because you need an origin of life event before you can have life that generates biosignatures that you go and you look for. And this was actually, I thought, really well illustrated by a paper by Affolter et al. In 2021, where they took Cassini data, in particular methane and hydrogen data from the plumes, to try to deduce the Bayesian probability that that was generated from metabolic activity or abiotic chemistry. And they generate and produce, they share their results as a function of the probability for an origin of life of an event on Enceladus. And unfortunately, we do not know that probability, but they emphasize that this is a key thing to understand before we can really start trying to deduce what worlds could have life. Because, as I said before, you need an origin of life event for there to be life. And we actually wrote a perspective that also emphasizes this. There's the possibility that ocean worlds, for instance, could be habitable, but were never conducive for an origin of life event. Therefore, they're habitable but uninhabited. Prebiotic chemistry is also very useful because it's good to help inform against false biosignatures. Abiotic organic chemistry in particular can get very complex. And you can imagine if you saw some of the things shown in this diagram, all of which have been shown in the literature to occur abiotically, you might be convinced that you found evidence for life. So going from left to right, we have the prebiotic milieu, so cyanide, formaldehyde, those sorts of things wouldn't be convincing for life, but we know they combine to form biologically relevant molecules such as nucleobases, amino acids. It's even been shown that the energy currency for life, ATP, can be generated abiotically. We can even form organic polymers of ribonucleotides, organic vesicles that could look cell-like. And it's been shown that these organic polymers can catalyze certain reactions. They can be encapsulated in protocells where they can still facilitate chemical reactions, but they're not technically life. So you can imagine if we go into the area of E, that could be very convincing, but it's still not life. And so it's really important to keep the complexity of abiotic chemistry in mind, because we don't really have a firm grasp on how complex it can get over geologic timescales. We just can't simulate that in the laboratory. We can't use samples on Earth because they're just contaminated by life, and it overprints any kind of abiotic organic chemistry. But it's really important to keep in mind for future missions that aim to look for life on other worlds. Perhaps we should be building instruments that can well characterize prebiotic organic samples. The problem with, of course, abiotic organic chemistry is that it's actually very difficult to characterize. And this has actually led to, I believe, a gap in mission work where there's a focus on habitability, and then we jump from habitability to looking for life on other worlds and biosignatures. And we don't really have a focus yet on missions to characterize the origin of life potential of planetary bodies. That's because it's just very difficult. I mean, we don't really know what conditions are needed for an origin of life event, but that doesn't mean that it isn't worth trying to develop hypotheses that we can test with planetary missions. However, this also goes back to what I was saying. They're just organic samples from the natural environment are just very difficult to characterize. So one technique that is great for trying to characterize organic compounds is high resolution mass spectrometry, or HRMS. This is different from typical mass specs because it has sufficiently high resolution that you can take the mass to charge ratio and derive potential chemical formula of your organic compounds. And you can combine this with fragmentation of those organic ions to get an idea of what the structure is. So you can really start learning more about the organics that are present. And this isn't just for samples that are returned to Earth for Earth-based analyses. We're actually going to be flying two high-resolution mass spectrometers in upcoming missions. First, the Dragonfly mission, as well as Europa Clipper. But even with high-resolution mass spec, it remains laborious to analyze these spectra, as emphasized with these 4 examples here. So even prebiotic chemistry in the laboratory, which doesn't even come close to approaching the natural complexity of a real environment, as you can see here with this Miller-Urey chemistry spectra, can get very complex. There's just no way that you can go through and analyze and characterize all of these organic compounds. And it's the same thing with extracts from meteorites, and similar to Titan-Tholen analogs generated in the lab. And even Cassini, we know based off results from Cassini's mass spec, which was not high resolution, but it still demonstrated that really complex large macromolecules can be found on Enceladus. So even with a high resolution mass spectrometer, those organic compounds would probably be very difficult to characterize. So usually when organic chemists are trying to characterize compounds, we take 2 different approaches. One is the traditional targeted approach. You look for products of interest. As I was showing in the previous spectra, you get a ton of organic compounds. So you have to pick and choose what you're going to characterize. And usually that is limited by what we have commercial standards for. And unfortunately, commercial standards are very biased towards Earth life because it is focused on pharmaceutical industry, for instance, and very focused on Earth life. And so because of that, we're actually ignoring a lot of organic compounds that aren't used by life. And so usually these kinds of strategies will look for amino acids used by life, the nucleobases, so on and so forth. But you're really missing all of that other data. And then there's the untargeted approach, which is looking for general trends. This is very common in the field of metabolomics. So for instance, it could tell you something about how does the aromaticity change from samples. It's really good for comparing generally different samples to give you an idea perhaps of how cooked a sample was compared to another sample, but it doesn't really give you specifics about the organics that are present. And so I'm going to be highlighting using a case study with the foremost reaction, the limitations of each of these approaches. And I'll begin with just taking a targeted approach. So taking a step back, why do we care about foremost reaction? So we know on early Earth and other abiotic environments, you can generate a lot of complex organic materials. But how do you go from that to something very organized like RNA remains uncertain. We know that today life uses DNA and proteins to store information and catalyze reactions, but the jump between complex organic chemistry and RNA is unknown. So it's been proposed that RNA might be too complex. Maybe there was something before RNA, a simpler genetic molecule, which we call a pre-RNA. But what that structure is also unknown. And for those of you who aren't familiar with the composition of RNA and DNA, they're composed of what we call nucleotides, which themselves have a nucleobase or a nitrogen heterocycle shown here, attached to ribose or deoxyribose sugar and a phosphate group. So the biggest problem with trying to form RNA under early Earth conditions is actually just getting the nitrogen heterocycle to play well with the sugars to generate the nucleoside. So without the phosphate, it's just called a nucleoside. And this step is very difficult. It's probably because in bulk solution, it's what we call a condensation reaction, which is you're generating a new bond that kicks out water. And so if you're doing this in water, it's just thermodynamically unfavorable. And in fact, previous work has tried to react these nucleobases with ribose under really optimal conditions. So high concentrations, drying it out to really drive the condensation reactions. And what they found is you get very little yield of the biological nucleosides with what we call the purine nucleobases shown here. And the pyrimidine nucleobases with the one ring nitrogen heterocycle shown here don't form any nucleosides. And this has led to the idea that pyrimidines are very unreactive with, in general, as are the purine nucleobases. But as I'm going to show you here, that assumption is wrong. So the foremost reaction is the polymerization of formaldehyde. So the repeated addition of formaldehyde, you have to have a little bit of some kind of higher order sugar like glycoldehyde, and you get more complex sugars. But when I've incubated the pyrimidine nucleosides, so thymine as an example here, one thing that you can see is that we have a lot of products. So in blue is just the spectra of the nitrogen heterocycle by itself. And black is the foremost mixture, which you can't really see because it's not ionized well using this technique. And then in green are all of the unique products. So if we zoom in here and we start looking at the chemical formula that we're getting and we subtract out the thymine heterocycle, we can get an idea of what the side chains are, which are very nucleoside-like side chains. They're not quite nucleosides, but they're very reminiscent of it. And so one thing that I found is that when I did this reaction at room temperature, frozen ADC, the reaction happens very quickly. And so these heterocycles are actually very reactive in these mixtures, even though past literature would suggest otherwise. But that's because they were very focused on looking for a particular product. So then I became curious, okay, well, how are these reactions even happening? This shouldn't be happening based off the past literature. So these are what we call isolated mass chromatograms. We take a particular mass to charge ratio, in this case, the 229, which has a chemical formula consistent with deoxyuridine. So a uracil deoxyribose nucleoside, it's probably not that, but it has that mass consistent with it. And we see when it appears depending on the reaction conditions. So as I mentioned before, when I have the uracil and I incubate it with the foremost reaction, we see a little bit of it. So that goes to show that it's happening very quickly. Even when I freeze it immediately at minus 80 C, and the only time I thought is to put it in the mass spec, we see some of that product forming. It forms also at 5 C after a week, as well as 30 C after 48 hours. But at 80 C, the reaction is not ongoing. So it actually doesn't like high temperatures. And then I also, of course, did a reaction with the nitrogen heterocycle incubated with ribose and deoxyribose to see if we are, if this is just due to some kind of mass spec chemistry, because you're ionizing these fragments, you could potentially form something from there, but it does not show up. So we know from past literature that it's not the sugar reacting with the heterocycle. It's been well established. But looking at the trends, I thought it could be, I deduced that it's probably happening through two different types of reaction mechanisms. Both are very similar. And they both involve the same first step, which is what we call the Michael addition. So the heterocycle represented by het here reacting with this carbon here to generate a 3-carbon carbonyl side chain. And then from there, you can get subsequent aldol reactions. So that's actually in the literature that you can get this acrolein to react with formaldehyde to form like this pentierythritol side chain. But I'm just saying you have a nitrogen heterocycle also attached with it. The other alternative is instead of formaldehyde, maybe you have a higher order sugar like glycoaldehyde. And so instead of getting a branched nucleoside, you're getting a straight chain nucleoside. And there's good reason to think that this reaction is ongoing in the foremost reaction because the foremost reaction generates sugars. And in fact, the reaction doesn't stop there. The sugars are then unstable under these conditions and they break apart. And that's actually what makes the foremost reaction turn yellow. When they break apart, some of the products that could come out based off the degradation of glucose includes Michael reactants like acrolein, crotonaldehyde, as well as their potential reactants that can form the Michael reactants, including formic acid and acetaldehyde. And so the next step was to do isolated reactions with nitrogen heterocycles to test which mechanism is ongoing. So first we have, these are once again isolated mass chromatograms consistent with a structure such as here, straight chain or branched. So uracil by itself as a control, it's not showing up. When we incubate it with just the acrolein to get the Michael addition, it doesn't show up. And when we add formaldehyde, it still doesn't show up. But when we have glycoaldehyde, the reaction shows. And when we have glyceraldehyde, the reaction shows a little bit. So what this is telling me is that instead of having repeated additions of formaldehyde, it's the higher weight sugars that are reacting to form more of a straight chain nucleoside analog. And in fact, when we compare the isolated mass chromatograms from uracil in a foremost reaction at 5, 30, and 80C, with that of the potential reaction mechanism, we see the same general trends, where it happens at 5C, 30C, but 80C, it's not conducive. But when we actually take these potential they have the same mass, right, as nucleosides occasionally. So for instance, here, this compound with the 268 mass to charge ratio has the same chemical formula as adenosine. But when we break it up, so this is a MS-MS fragmentation profile, we can see that it does not match adenosine, a control. And so in previous work, if they were looking for adenosine, they would say, oh, adenosine isn't formed in this foremost reaction. Adenine's not reactive. And they don't bother to characterize this nucleoside analog because it's not interesting to them because they're taking a targeted approach, but also because it's very difficult to characterize this. So these are a potential structure that I've drawn here. I can't actually say this is for sure the structure because I don't have a commercial standard for all the possibilities. This is actually a big hindrance for prebiotic chemistry because, as I mentioned before, most of the organics you're generating abiotically, we just don't have standards for. And in fact, if you go into other previous work, you'll start seeing this trend actually where the nucleobases are more reactive than we've previously given them credit for, but they're just not forming necessarily the things of interest. So here's a paper by Becker et al. In 2016. And they were showing, they were incubating diaminopyrimidine with the foremost reaction to generate adenosine. This is an isolated mass chромatogram of something of a mass that's consistent with adenosine. And what you can see here is actually there are quite a few peaks that have the same mass as adenosine, but the retention time is different. They don't actually match up with either adenosine shown in in green here, or its potential isomer, where it's instead of a furan ring, you have a pyran ring. So that's shown in the red and the green here. You actually get all these other adenosine-like molecules, but they don't talk about it in the paper because you can't characterize them. So targeted approach, I hope what you get from this is that it's very limited. It depends on what you're actually looking for. So what you don't look for, you'll never know if it's there or not, especially if it's not even reported in the paper. And it's very biased towards biologically Earth-centric molecules. And so one of the things I took away from my work is, are these nucleoside analogs actually common in prebiotic reactions, common in the solar system? We found that Michael reactants are thought to actually potentially be a source of amino acids in carbonaceous chondrites, for instance. So these reactions could be ongoing in those. They could be delivered elsewhere. And most importantly, if you saw a nucleoside analog with during a planetary mission or with return samples, could it be mistaken for a putative biosignature? So let's go a different direction. Let's go an untargeted route and see what kind of information we can get from that. So previous work has studied and characterized the foremost reaction using either Van Crevlin plots or Kendrick maps. These are kind of the gold standards for an untargeted approach. So Van Crevlin plots are just when you take the hydrogen to carbon ratio and plot it against oxygen to carbon ratio for each of the products identified based off their chemical formula. And what you can get from here, so this is the foremost reaction, and they've plotted it against Murchison, an oil sample, and E. coli. And basically, when all the points are on the left-hand side, it shows that it's probably been really cooked. It's very aromatic. You don't have a lot of reduced organic compounds or compounds with other atoms like oxygen, whereas the foremost is more diverse. So that's the kind of information you're getting. The Kendrick map is you choose a molecular fragment of interest and you take all of the products identified and you're looking at the difference between that mass. And so these are all the products identified along the horizontal axis, and then they separate based off their difference from the molecular fragment. And so what you get is horizontal lines. Peaks that fall along the same horizontal line have the same, they belong in a homologous series. So you can imagine if you had adenine with a methyl group, then next to it would be adenine with two methyl groups and so on and so forth. So it can give you an idea of polymerization reactions that happen, but nothing specific, nothing about the actual specific compounds. So I hope to have emphasized using the foremost reaction as a case study, the limitations of both of these approaches. However, there's a third approach that we can take with machine learning to try to not be able to necessarily say exactly what organic compounds are present, but to actually get a better idea at least of what their structures might be. So something in between a targeted and an untargeted approach. And in fact, machine learning is critical for planetary missions and for the characterization of organics, because even when we fly high-resolution mass spectrometers, such as with Europa Clipper, they generate really large files. We're talking gigabytes here, and that's already difficult enough to parse through on Earth. But when you have a planetary mission, you're limited also with how much data you can send back with any one pass. So with mass specs, it could require multiple passes to send back data from one experiment, and that can really hinder missions. And so we need some kind of way to autonomously pre-process the data to get back what's really important back to the scientists so they can make informed decisions. And in addition, it's also critical for return samples because we can use machine learning to really tease out a lot of information we wouldn't otherwise, that could be like left on the table. And when you're really limited on sample mass, that could be really important. And then finally, as I've brought up a million times, this is my soapbox, it can be critical for abiotic studies because we just don't have commercial standards. So even if we can't ever say, like, narrow down the exact specific structure, perhaps we can at least say something about what it could potentially look like. And so now I'll go into strategies I've developed to facilitate the characterization of high-resolution mass spec data using chemometrics and machine learning. In particular, I'm trying to develop a pipeline that can facilitate the autonomous pre-processing and reducing dimensionality of high-resolution mass spec data sets. So the first thing that I've done with the script that I've built using R and Python is that you give it an open source format of high-resolution mass spec data, and what it'll automatically do is cut the entire spectrum into really narrow spectral windows. And then from there, what it's doing is it's trying to fit a function to the peaks here, to the curves. And the function in particular that I'm trying to fit is a pseudo-Voigt function, which is just a combination of a Gaussian equation and a Lorentzian equation. So Gaussian, the peak's a little bit fatter, Lorentzian, they're a little bit more narrow. This is what the actual equation looks like. And each equation, both Gaussian and Lorentzian, has certain parameters, in particular full width half max of each peak, the peak center or the mass to charge ratio, its intensity, and then overall the proportion of Gaussian to Lorentzian. And what we're doing is trying to fit this equation to get these parameters for all of the peaks present in the mass spectra. And what we can do to do that, actually, you have to have a starting guess and bounds for upper bound and lower bound for each. So usually when people go through this, they might go and look at the spectra themselves and manually decide what is a good guess, manually decide what's a good upper and lower bound. But you can do this autonomously if you use a combination of first and second derivatives to get good estimates. And that's what I've done. And then finally, we take our upper bounds and our starting guess, and we optimize the equation using a Levenberg-Marquardt optimizer. And one of the things that I found is that this is very sensitive to what your starting guess is. And the reason why it can be visualized with this diagram. So if you can imagine that each point here represent a different starting guess of all the parameters, if you started somewhere like here, you could easily find the global minimum. This is a very good match. But if you start over here at A, you could get trapped in a local minimum and it optimizes, but the fit isn't good. And so because of that, I've had to have the script be very iterative in its starting guesses and then take the global, the best fit essentially from there. And what I found is that it works really well with mass spec data. So this is just kind of demonstrating how well of a fit you can get with mass spec data, even when it's in the noise, low intensity, 300 arbitrary units, it's able to capture the peaks. If it's a single fat peak, it's able to get it. When we have overlapping peaks, this is when it's actually critical. It can also deconvolve those peaks. And what it's doing is at the end of it, you get all these peak parameters, and then also from those peak parameters, integrated areas for each. And since this is high resolution mass spec data, we have an accurate mass to charge ratio, and then we can start predicting chemical formulas. And we can do that using a package called pulp in Python. This is a linear solver to determine the chemical formula. And this is actually follows the same rules that if you have mass spec software, this is what it's also doing. But when you have the software, first of all, it's not open, it's not free, but also you can only do one peak at a time. Whereas what I've done is have the pulp package look at all of the peaks that are gathered and then make be able to solve for what could be good solutions for chemical formula for each. And essentially the way it works is you give it an upper lower bound for the number of each element. Same thing with the mass spec software. And then it uses the accurate masses of the elements and the mass of the proton, because this is protonated ions, to determine a good solution. What minimizes the observed mass, the difference between the observed mass to charge ratio versus theoretical mass to charge ratio that it's trying to fit. And so this is what a typical output looks like. So you have your observed mass, theoretical mass, the error, and then it has the total number of, in this case, carbon, hydrogen, oxygen, nitrogen. And then because I'm doing experiments where I know I'm only interested in products that are not present in any of the controls, so I told it to kick all those out and only look at things that aren't in the controls. I think that these are probably all the nucleobase adducts, so the nucleobase plus a side chain. And so we can subtract the chemical formula of the nucleobase to get the chemical formula of the side chain. So that's what these are representing here. And then once we have the chemical formula of the side chain, you can make some general rules to get an idea of what kind of structure this is. Is this consistent with a carbonyl group, a sugar-like group? And that's also what I've done. So now I'm going to go ahead and apply this to the foremost reaction to demonstrate its utility. So this is adenine, the results with adenine in the foremost reaction. And this includes data from the reaction done at T0, so minus 80C frozen and then thawed, and then incubated at 24 and 54 hours at 30C. And what you can see here is the distribution of potential side chains. And keep in mind, this is an absolute distribution. It's not absolute abundances, it's relative abundances. But if we assume that the heterocycle is ionizing, is what's actually ionizing, then we could say potentially that these are kind of on the same playing field. So what's really stands out to me is that there's a lot of these carbonyl side chains forming, and it's regardless at how the condition that it's done at. So T0 versus 54 hours, it happens very quickly. But we also see these sugar alcohol side chains, which I had mentioned earlier. So that's like a nucleoside that has an extra OH group. And what I really want you to take away from this is that it reduced a spectra, spectra that had 326,201 peak candidates to about 2,200 unique peaks with elemental formulas, the deconvoluted peak areas, and that were isolated from, that didn't include things from the control, so they're truly just adenine adducts. And so it's a reduced set, but it's much more information rich. So this is one way we can reduce our high resolution mass spec data. Another thing you can do is when you start taking, so I took all the other nucleobases that I studied, and I wanted to see the distribution of sugar-like side chains by how many carbons they have. And so what you can see here is each of the nucleobases divide with the sugar side chains graphed by how many had 6 carbons, 7 carbons, and so forth. And what you can see here is, with the exception of 5-hydroxymethyluracil, all of these nucleobases tended to form sugar-like side chains that were 6 carbons long. And then you can also plot by, the temperature or the duration. So we can see that these reactions are happening very quickly at T0. So it's just another way to explore your data very quickly. So in conclusion, machine learning can facilitate the analysis of complex mixtures by essentially giving you an informed, untargeted approach, right? So instead of going in and saying, I want to look for these specific ions, I took an agnostic approach, look for all of the peaks present, tell me which peaks are not in the controls, and then figure out the chemical formula from there. So it's a very agnostic approach, but instead of completely untargeted, we're still getting some information about the reactivity. And so another question that I wanted to ask is if we could use machine learning to help learn more about the structure of the identified organics, because you can only get so far with the chemical formula of the side chains. And so for this, I was awarded some money through JPL to develop a machine learning model that leverages mass spec fragmentation patterns that are available on open databases. So they're actually all metabolomic databases. And they have hundreds of thousands of MSMS fragmentation patterns. And I wanted to build a classification model that could take that input and then, based off the fragmentation pattern I give it, tell me what type of organic I'm looking at. So it's not the exact organic compound, but so for instance, if we use an amino acid as an example, whether it's an amino acid, a nitrogen heterocycle, a sugar, but we could also potentially even get more specific. Is it an alpha amino acid, a beta amino acid, or a gamma amino acid? And so it can start giving you more information about these organics, which we don't have standards for. And so the way that this works is, first, I use an unsupervised learning to learn associations between the mass fragmentation peaks using an algorithm called spec2vec. And that's actually based off a language model called word2vec, which might be easier to explain how it works using word2vec. So word2vec, you have a series of words. Let's say you have a book. It's looking at how often all the words co-occur to determine how related they are. So for instance, you could imagine king and man are going to occur more often together than king and woman. And this is demonstrated by the resulting vector that's output. So you can see the colors here between king and man are more similar than between king and woman, whereas man and woman would be more similar than woman and king as shared. And so with mass fragmentation patterns, what this is doing is, instead of words, it's looking at the difference between the fragmentation peaks from the parent molecule. And that's important to note because we don't want to look for exact matches, exact fragmentation peak matches, because you could imagine you could have something like, let's say, adenine versus methyladenine. It's only different because it has an extra methyl group. So that extra methyl group could create fragments that are each like an extra methyl heavier than the original adenine. And so because there's actually not a lot of overlap with the fragmentation peaks, then you would say, oh, they're not similar at all, even though it is almost the exact same structure, just with a methyl group. And so learning the differences from the parent molecule and seeing how often those fragment peaks occur together is one way to get around that issue. And then what the algorithm does is it's essentially, if we wanted to visualize how it's looking at the shape of the vector that comes out from this. So if we were to plot all of the peaks here and the differences, the vector might look something like this. So they're not exact, right? So for the instance, with the case of adenine, this could be adenine and this could be methyladenine. They're not exactly the same. They don't perfectly overlap, but the overall shape, so the overall difference between each of the peaks is similar. And so therefore we know their structures are similar. And then we use that data, that relationship information, and we build a classification model using gradient boosting. So you can think of this as, if you're familiar with random forest, it's like random forest. It's decision trees that are learning as it essentially goes. And importantly, for any classification model, you need a label. You need a labeled data set. And in this case, using metabolomic databases enables us to use the labeled data set they have there. All of the entries actually have an organic hierarchy. And it's so for instance, is it an organic or inorganic molecule? And then the next structure might say, is it a heterocycle or an amino acid? So we have that labeled data, and it can go down to be more narrow, like alpha, beta, or gamma amino acid, and then eventually ending on the exact name of the compound, like glycine, for instance. And that classification label, if you're interested in learning more about it, is generated through an algorithm called classifier. And so you can learn more about it by going to Fanong et al. 2016. But it really helps. Without that kind of label, this work wouldn't be possible because there's just so many organic compounds. And what I'm showing here is the first initial attempt to see how well a classification model did. So we have our different groups here on the side. So these aren't all the groups. I had to cut it off because it just gets really big. And this is what we call a confusion matrix. So it's a way to evaluate how well your model did. What you're looking for is this diagonal. You want it to be very blue or have ones, because that means it would be a perfect model. Anything that falls off of the diagonal is where the model failed. So in this case, the model actually failed a lot in misidentifying the category amino acids and derivatives. It kept identifying other compounds under the amino acid and derivative when it wasn't. And I think that's probably because that's a pretty broad category, and I would need to look more into it, but the and derivative makes me wonder what is included under that, and it's probably confusing the model. And one way to that demonstrates what I mean by this is if we do a t-SNE. So this is showing essentially, it's plotting how related all of the different inputs, so all of the MS fragmentation profiles I put in, how related they are according to the spec-to-vec algorithm. And so we would expect that if they're going to be, if the spec-to-vec algorithm input is going to be great for building a machine learning model, we should get nice clusters like we do here, right? Which we do. We do see some nice clusters. But if we look at the aminosaccharides shown in red here, for instance, we're actually seeing multiple clusters. And that's probably what that's telling me is that's because it's a very broad group and there's some diversity within amino acid saccharide, or sorry, within the aminosaccharides. And it would actually be very difficult probably to come up with a model that can accurately say something is an aminosaccharide. But because they are forming nice clusters, perhaps it's actually easier to be more specific. And so I decided to look to see if it'd be easier to classify at a lower taxa. So for instance, instead of saying something's an amino acid or not, tell me, is it an alpha, a beta, or a gamma amino acid? And that's actually even more informative. So in this case, because I love nitrogen heterocycles, I used different classes of nitrogen heterocycles to see how well the model would be. And it actually improved the model. So instead of 80%, now it's 86%. And so this tells me that there is promise for using this strategy for getting more details about these organic compounds we're detecting and characterizing using mass spec data. And so overall, I've hoped to emphasize that there's two approaches we tend to use: the targeted approach, which is very biased towards Earth life and what we have commercial standards for, but you lose a lot of information because the majority of the sample is unfortunately not Earth-centric molecules. It's compounds we don't have standards for. And then there's the untargeted approach, which is great for trying to get an idea of general chemical trends, but it doesn't give you specifics about the organic compounds. Whereas chemometrics machine learning can provide a way to do an agnostic search, an agnostic characterization of all the compounds present and actually start getting at more details about the reactions that were ongoing and who is there, even if it's not the exact compound identification. So in conclusion, prebiotic chemistry is absolutely critical for life detection missions. It's important to be able to deduce the origin of life potential of other worlds, because if it didn't have a good origin of life potential, then that tells us that it's probably never had life. It's also important to be able to understand abiotic chemistry because it can also look like life. Unfortunately, abiotic chemistry is very difficult to characterize, even with high-resolution mass spec, but we've talked about this third approach using machine learning. And this could be important for future proposed mission concepts such as Mars sample return, Ceres sample return, and so forth, as well as ongoing missions such as OSIRIS-REx, analyzing those samples, as well as Europa Clipper and Dragonfly, each of which have high-resolution mass spectrometers. And with that, I'd like to thank my past collaborators and advisors, specifically Lori Barge, Christopher House, and Michael Callahan, and my sources of funding. And now this is when we take a little bit of a right turn, and I start talking about the work that I proposed to do at UT in this coming year. So I'm very interested in understanding whether subsurface environments could generate conditions conducive for the origins of life. And by subsurface, I mean like the deep sea or deep crust, where you're not exposed to UV chemistry, wet-dry cycling necessarily, or spark miliary spark discharge, or even exogenous delivery of organic materials. And under such conditions, what's been proposed as a probably important for generating organics in these environments is Fischer-Tropsch type chemistry. So this is where you take CO2 or CO, hydrogen, and a metal catalyst under high pressure and temperature, and you can generate methane and organics. And it's been proposed that the hydrogen could be sourced in these environments from serpentinization, which can happen at the seafloor or deep subsurface environments. Unfortunately, previous experiments that have demonstrated this feasibility were actually found to be wrong, in the sense that it was found that the organics and methane generated were actually all organic contamination from the olivine mineral that they were using. And this was shown using when you actually use labeled CO2, you don't actually get labeled products. And so it's been suggested then that this reaction, although thermodynamically should be favorable, is kinetically inhibited in bulk solution. And that could have potential implications for where the origin of life happened on early Earth. Perhaps it couldn't happen at hydrothermal vents. Perhaps you do need a subaerial pool where it's exposed to air and wet dry cycling. This also has implications for ocean world being able to host life, where like Enceladus, for instance, it's any kind of organic chemistry is happening in the subsurface environment or the crust. And so it's actually been a growing opinion that these ocean worlds might be habitable but uninhabited. However, there's a paradox between that experimental work and field work, where people who have gone into the field have found where there is serpentinization ongoing, there is the generation of fluids that are enriched in hydrogen and methane, and isotopic studies of this methane suggest that it is actually abiotic. So what's going on here? Well, one potential solution is olivine fluid inclusions. So as I mentioned before, past Fischer-Tropsch type experiments trying to do this in bulk solution was actually releasing organics that were trapped within olivine. And so this also suggests, well, if you think about it, where did those organics come from? And in fact, previous work has looked at olivine fluid inclusions and has suggested that these could actually be one of the largest reservoirs for methane on Earth. And Lemadrid et al. In 2017 actually found that when you synthesize fluid inclusions in olivine and study how serpentinization is ongoing there within the fluid inclusions, you get really interesting chemistry that you don't get with a bulk solution. So within the fluid inclusions, because it is a small and semi-closed system, as serpentinization is ongoing, your fluid is actually getting enriched in salts. And that eventually slows down the serpentinization. And then over time, the salty fluids percolate through the rest of the olivine crystal and it becomes more fresh. And then when it becomes more fresh, serpentinization picks up again and it gets salty. And then it's the same cycle goes on over and over again. And so this is one way to get high salinity, low salinity cycling, or high water activity, low water activity cycling, which is basically what wet-dry cycling is doing on land, and what's been invoked as really important for the synthesis of organic compounds through condensation reactions. So if you recall, I said the nucleobases plus the sugars don't like to form the nucleosides in bulk water because that's a condensation reaction. You're generating water in a water solution. And so having this kind of chemistry within fluid inclusions could provide a way for not only reducing CO2 to generate organics in subsurface environments, but also potentially forming more complex organic molecules through condensation reactions. However, a potential reaction mechanism that could inhibit CO2 reduction is the fact that when you have carbon CO2 in fluid inclusions, it's been found that you get carbonation. So the formation of siderite or magnesite with the iron and the magnesium in solution rather than CO2 reduction. And so my work with the UT group is trying to look at what is the balance between carbonation and organic synthesis. At what conditions can we actually get CO2 reduction? And so this is divided into a 4-step plan. First, I want to establish a baseline and a protocol for analyzing olivine fluid inclusions. So I've been working with Dr. Anders Fall from the Synthetic Fluid Inclusion Laboratory at the Bureau of Economic Geology at UT. He has a ton of experience generating synthetic fluid inclusions. And so what we're doing is first trying to develop a good protocol where we don't contaminate our fluid inclusions with organics from, various sources. And then also seeing how well our analytical protocol is at characterizing the fluid inclusions, but also just characterizing what contamination is already present before we add a source of carbon. And the fluids that I'm using is actually a simulant for earlier, so anoxic, ferrous-rich, salty, and a pH of 7. And I've listed here some of my other collaborators with this project. So I'm using TOF-SIMS that can help get an idea potentially if there was CO2 reduction, but it can't really tell us too much about organics and who's there because it generates a lot of fragments and can be difficult to analyze. It's also very slow. It's a pretty slow analysis. We have MALDI mass spec, where that's actually going to be using a high resolution mass spectrometer and a laser system to ablate materials to look for organics, and we can couple that with fragmentation so we can start learning more about the organic materials. Unfortunately, MALDI mass spec is not really used for geological samples, so that's going to require some method development to see how well it does at getting into and ionizing the organics within the fluid inclusions. And then Raman we can use to monitor the reactions as they're ongoing, so we can see hydrogen peaks, methane peaks, and so forth. And then GCMS, I'm working with Dr. Tongwei Zhang, where we have a ball mill that can crush up the olivine crystals and then suck up the gases that are released and characterize them using mass spectrometry. So task 2 would then be looking to confirm MALDI's utility for characterizing organics. And so for this, I'm going to have the same fluid sample as task 1, but I'm going to dope it with adenine. And that's because adenine is very ionizable for mass spec. It should be easy to pick up. But another reason why I want to do this is I'm curious to see what happens to the adenine when it's subjected to the high pressure and temperature to make the synthetic fluid inclusion. It's probably going to break up. But as I mentioned before, the fluid inclusions generate these conditions that are really promising for more complex abiotic reactions. So it's not just interesting for CO2 reduction. So if we want to do future prebiotic experiments using synthetic fluid inclusions, it's good to know what's going to happen with these starting organic materials. And then task 3 is actually testing CO2 reduction in the fluid inclusions using various carbon sources, such as carbonate. If that doesn't work, we can use oxalate, which breaks down into CO, CO2, and H2. And we can do these both with and without nickel, which can really help kickstart the reaction. And then finally, I'm working with Dr. Elizabeth Spears and Krista Soderlund to develop strategies to model any organics or volatiles that we generate in these reactions, to model their transport in icy ocean worlds, because the experiments can only tell us, if this is possible, but the modeling can help inform how much this could influence the origin of life potential of these planetary bodies, and also what we could look for in future missions or in past mission data to see if these reactions are ongoing. And with that, I'd like to thank you for your time and attention, and I'm happy to take any questions.
Thank you, everyone. Yeah, if anyone has any questions, then you can raise your hand or you can feel free to drop them in the chat. Well, looks like we may not have any questions.
Shocking.
Yeah, the danger of a Zoom seminar, unfortunately.
Okay, I'll ask one. So what are the temperature conditions are you thinking about for your experiments?
Yeah, so there's 2 different conditions. There's the conditions that we expose the olivine crystals to generate the synthetic fluid inclusions. And then there's the conditions to actually get serpentinization ongoing. So to generate the synthetic fluid inclusions, I think it's like something like, I'm trying to remember, 700 C and 2000 bar, something like that. Whereas serpentinization, I'm probably going to do it at between 250 and 300 C at one bar. When you heat it up that much, the pressure and the fluid inclusions can get pretty high, the internal pressure. So I think about like 500 bars. So we don't really need to expose those to high pressure at that point.
Do you control the redox condition?
So I've had it so that I prepared the solution so that it's anoxic. And then unfortunately, just when you have to prepare the fluid inclusions, we have like these platinum tubes. We put the olivine crystals in and then the anoxic fluid, and then we seal it. But of course, during that process, it is exposed to oxygen from the air. So we also added titanium powder as a way to pull down any oxygen because it reacts with the oxygen pretty quickly to form insoluble titanium oxide, so they won't get trapped in the olivine fluid inclusion. So that's my attempt to control the oxygen.
For both set of experiments, for synthetic melt inclusion and then later addition.
Yeah.
Okay. Thanks.
In the chat, we had one person ask, what is the dimension of the Levenberg-Marquardt optimization?
I'm not sure what that question means. What is the dimension?
I'm guessing the number of parameters in the state that you were running over.
Okay, yeah. Let me think. So we have full width half max, the peak center, proportion Gaussian to Laurentian, peak intensity. I think that is about it. So like four parameters. But it's for each peak within the spectral window. So that's 4 parameters for peak, depending on how many peaks we predict are present based off using the first and second derivatives. So it can, at minimum 4, could be much higher than that if you have more peaks in the window.
All right, any final questions before we break at the top of the hour? Well, if not, Laura will be here as a visiting scholar, so please feel free to reach out to her. But thank you very much for the presentation and for telling us a little bit about your work. For everyone else, as a reminder, CPSH, we're going to have a collection of what we're calling lightning talks on Monday, the last day of classes. So we look forward to seeing you there, and you should see an announcement on that here in the coming days.
Great. Thanks, everyone.
Bye, everyone.